Hello, welcome to Do-It-Yourself Meta-Analysis and Forest Plots in Less Than an Hour. I am your host, Dan Dick, and we will be going through this today. So first we'll talk a little bit about what meta-analysis is and how to set up the beginning parts of it, which includes framing a clear question and then generating a Prisma checklist. So in every presentation about meta-analysis, I like to include a slide like this. Just basically, why do meta-analysis? So as we know, field trials are expensive and also messy, and studies within field trials can often disagree due to the climate, the differences in soil, differences in cultivar, or methods of application. So really, we understand that no single paper gives us a full answer to our questions. Meta-analysis is a way to combine the results of many studies to estimate a true effect size regardless of climate, soil, cultivar, or method. So in horticulture, we know that crop responses are highly variable, and by including multiple papers, meta-analysis strengthens our recommendations to growers. So an example would be the pooling of 10 studies of different cytokinin sprays can give us a single fruit size estimate when you have individual studies that might not come out in the same way. So what is a meta-analysis? Really, it's a systematic review and a quantitative synthesis. So there's two parts of this. The part one is the systematic review, which is simply a search for all of the relevant studies that cover the topic and have information about the thing you're trying to understand. And through this, we can apply a strict inclusion or exclusion criteria such that papers that do not fully meet the scope of the study can be excluded, while those that do are included into the meta-analysis. The second part is after you find all of the papers that are included within your systematic review, you extract numerical data, which includes the means, the standard deviations, and the number of observations. And then that allows you to calculate the effect size for each study. The combination of effect sizes from each study are combined into one pooled estimate, which is the overall effect size of that topic. So the goal is to get a single best estimate and confidence interval from noisy papers. So really you have to form a hypothesis before you can even start to look for research papers that have the information. So a very simple template that I came up with was just, does treatment change trait in crop compared to controls in field trials? So the demo question for today is, do pre-harvest cytokinin sprays change blueberry fruit size compared with unsprayed controls? So a Prisma diagram is a word that you'll see in the book Meta-Analysis with R, and Prisma stands for Preferred Reporting Items for Systematic Reviews and Meta-Analyses. And the basics of it is that it shows how many papers were found in your searches, how many were screened for eligibility, and then it has the exclusion and inclusion criteria for those papers. This is required by many journals, including the Frontiers in whatever, like Frontiers in Plant Science, like the journal that I have submitted and gotten my publication for the meta-analysis in. And what this does is it really adds transparency, and it shows that you didn't just cherry pick your results. Sometimes you can exclude papers based off criteria that really shouldn't be exclusion criteria. So what this does is it allows the reviewers and the readers to see how many papers were included and excluded from your results. What it gives you is a super nice PDF or PNG file that has the Prisma diagram, which is what I'm showing here. So in the case of the example, I searched 4 databases. From those databases, I immediately removed 4 papers because they were duplicated, and 4 papers that were removed because they were not peer-reviewed and included in conference proceedings. So before you do any exclusion of any papers or publications, you should sit down with your advisors or try to come up with reasons for not including criteria. In my example, duplicates obviously are just duplicated, those should be removed. But conference papers typically are not peer-reviewed, and that's not a problem, but the problem is that the same data can appear in more than one publication, since Conference proceedings typically are held every year or every other year, and if a paper has multiple years of data, sometimes they show only the pilot or the first year of study in the conference paper, and then they publish multiple years in the actual publication, which is the reason why those were removed. So once you have completed the Prisma diagram, and you know how many papers should be included in your paper, then you need to walk through the extraction of the data from these papers. And I'll show you how to do that now. Okay, so for our example, I'm on Google Scholar here, and I just typed in cytokinin blueberry with the minus symbol and then a word that I don't want to find in the results, which in my case is micropropagation. So you'll have to discover what words you want to exclude from your search results. For my example, if I don't exclude this and I search, all of my results will be about micropropagation or leaf regeneration, things like that. So I don't need this word in that search result. So once again, if I type in the minus symbol and propagation, it will negate all of the results that included that in it. So from here, I'm just going to go down, here's my paper here, go down to this paper here, and I will run you through how to figure out if the paper has the information that you're looking for. So in my example, I was looking for the increase of fruit size or decrease or change with cytokinin in blueberry. So this paper titled Bioregulators Can Improve Fruit Size, Yield, and Plant Growth of Northern Highbush Blueberry. And I know just from having this paper in the past that it does include cytokinin, so you'll have to figure out, sometimes the paper might not include the actual name. Like in our case, BA is referring to cytokinins, so you have to really understand what acronyms are used for the different treatments. So from here, you can kind of skip through and figure out what was actually applied. So I'm looking here at the abstract. I see that they have 100 ppm BA and untreated control. You can read down here. NAA and BA treatments generally tended to increase the size of blueberry fruits. Okay, cool. So I know that they're going to include some data about the fruit size of blueberry fruits. And so from this, I really know that this paper does include applications of cytokinin, and it also includes the results of increasing in fruit size. So from here, I'm going to look through for a few things. The first thing is basic plant material information. So how old were the plants? Okay, I see they were grown in containers. I can note that the cultivars were both Duke and Blue Crop, and the other stuff is just pretty standard. From here, I can figure out what they applied. So look for the part that says BA. So they applied BA at corolla drop, which is when the flowers are starting to senesce, and they applied it simply at 150 ppm. When we scroll down, we should look for either a table or a figure that has the information. And in our case, it's the first table here that has the information that we need. So note that we have two different cultivars. We have Duke at the top. And then if we scroll over, we have, maybe it's a different table. Yeah, a different table here. Blue Crop is the second table. So within the first table, we need to look for our information. In our example, we have to write down the results of control and the results of the BA treatments. In my case, I know that there's not enough observations to have a split between the two different ppms of BA. So in this example, we will be combining the results of both of these two cytokinin treatments. And how to do that is really you look at what parameter you're trying to look at. In our case, it's average fruit weight. It's in grams, so note that if you have a different paper and a different metric or a different unit, then you have to convert that. This might be in milligrams, this might be in kilograms per plant. And so whatever it is, you have to figure out what that unit is going to be. In my case, it makes the most sense to use grams because that's how a lot of papers report differences in fruit mass. So how to do that, you look at the means across both seasons of the control, and you would average those two. So in our example, I'll bring up a new Excel sheet, a column that is study label. And in our case, this paper, I'll call it Millich 2018 for now because that's the name of the paper. And here I'm going to have a column called, let's call it mean E, SD, E, mean control, SD, control, and then number of observations for experimental and number of observations for the control. From here we can write down these two means here. So the first one for control would be 1.48, the second one 1.4. The average of these two is 1.44, so we can go ahead and replace that value with the single value because we're averaging across both years. The standard deviation is already listed here, so we can average those two. In the first year it was 0.4, in the second year it was 0.23. So once again, 0.4, 0.23. Find the average of these two. In our case, it's 0.315. And so now we have the mean and the standard deviation of the controls. Now remember that it's across two different cultivars, so in the case of differences in cultivars, if we're averaging between these two, once again, we would put all of these together, but for our case, we're just going to look at the one here, just for simplification. So the second part is to average all of the controls together, or I mean all of the treatments together. So in our case, we have 4 of them. We have BA at 50 ppm in 2026, 2027, and then BA at 100 ppm at 2026, 2016, and 2017. So we can go ahead and average those, 0.85, 1.7, 1.96, and 1.87. From these 4 values, we get an average of 1.845. And then once again, we can delete those and then do the standard deviations, which in our case would be 0.35 and the other ones. So 0.35, 0.18, 0.34, and 0.2. The average of these is 0.2675. And so now we have all of the means and deviations for these. But then the last step is to do the number of observations for both the experimental and for the control. So how to calculate that? It's pretty simple. So the number of observations is how many times did they apply something or not apply something, and write down a separate measurement within the same study. So in our case, if we're just looking at Duke, a single cultivar, there would be 2 observations for the control with an application in 2016 and a second application in 2017. So for the control, we have 2 observations. For cytokinin, since we're grouping the 50 ppm and 100 ppm together, we have 4 observations. We have 2 in 2016 and 2 in 2017. So the answer here would be 4. So this is how you would fill out each individual publication with its mean, its deviations, and its number of observations. So these are the standard Excel sheet templates. When I ran through the example, I had a slightly different name for the columns because I wasn't looking at this, but you can see by the column name and the description what each of them means. So as I've gone through, the study label is the author year of the paper. The N is the number of treated samples or control samples. The observations for each. The mean values are the means of either the treated or the control group, and the SDE or SDC are the standard deviation of control group and standard deviation of treated groups. You can have other columns I didn't include in our example, such as what hormone was applied, a subgroup, or a species. This is not necessary if you do all of your parsing in different sheets. This is if you had a big long sheet with all the different studies within the same sheet, you could separate them out based on the subgroupings. So as I mentioned in the walkthrough, to calculate the number of observations, it represents the total number of independent trial observations. So the formula is n is equal to the number of distinct treatment applied multiplied by the number of seasons and the number of locations. So if we had more than one location across both years, we would have to have a multiplication factor based on that. So an example would be if you have 3 treatments applied in a study, and it was conducted over 2 growing seasons, and data was collected from 2 different field locations, and all of those field locations were put down as separate means in the paper, then we would multiply each of those together. So it would be 3 times 2 times 2, which equals 12 observations. Do not count the number of plants. That's not an observation. That's simply part of the setup of the trial. And it's only the number of actual mean values that are presented and where those means come from. So like I've ran through, this is an example Excel layout with one row per study comparison, and make sure that they're all in the same units. In our example here, it's grams per berry. Okay, so now that we're on the R example markdown that I made, I'll walk you through how to do this. So the first thing I need to do is to load all of the packages that you will be using for this meta-analysis. So this is a very simple thing. It just checks if you already have something installed. It won't install it, but it will if you don't have it. So in our case, we're using dplyr, we're using the meta package, and we're using read Excel just to read in the Excel file. And the bottom thing here is just a command that sets the figure width and height so that we don't have to adjust that when we're saving each individual file. For this, I'm going to simulate some dummy data using 10 different studies. And the studies have, you can see here the number of observations are, between 10 and 20 with an average of 10 for the experiments, and sense control typically has less. It's between 5 and 1. The data here, I'm forming a data frame that just has the study name pasted between study 1 and 10. I'm inputting the 2 different number of observations, and I'm computing randomized means within a normal distribution. Same thing with the standard deviations, and I'm giving myself a hormone and subgroup classification based off of the paper names. So I'm going to run this. To prepare your data, you have to have a new column that's called mean difference. So the mean difference is simply the difference between your treatment group and your control group. So in order to do this, you just take your data frame, you pass it through a pipe operator that's supplied with the dplyr package, and using the mutate function, you make a new column called mean diff that is equal to the column mean of experiment minus the column mean of the control. And just so we can understand how that looks, I will show us data now. So now we have study label, we have number of observations, we have the means of the papers, standard deviations, and then we have the mean difference as a new column that is computed as the difference between the control and the experimental group. So now we're getting into the actual meta-analysis. Now I've made nice labels for what each of these are doing. Very simply, this is laying out how the result is being read. So the result is something that gets fed into the meta-analysis package using the metacont function. And metacont needs to know the names of the columns within your data frame. So very simply, it has these defaulted names of the columns, and I've kept the names the same as they're listed in the function. So in our case, the number of observations for experiment is the same name in the data frame. It's n.e. Going all the way down, there's a few things that you have to adjust. So this is the combined fixed or combined random effect. So you'll learn more about this in the book when you read it. But the difference between a fixed and random effect is that a random effect does not assume the same heterogeneity between studies, while a fixed effect does. So since we cannot assume that the uh, standard deviation should be the same between studies, we use a random effect. To combine the random effects, this is how we get the overall effect size. What this is doing is it's combining all of the studies together so you can achieve a single effect size. This here is the method used to create that overall effect size. In my case, we're using mean difference because the mean difference is tied specifically to a unit. So for our examples that we've been running with, we have up here, this is all in grams. So since each one of these observations is in grams, we can directly compare them within a unit. So because it's within a unit, we can use mean difference. If they were different in units, you can use something called standardized mean difference, which will get rid of the units and do proportions as a percentage change. But I found that by using mean difference, it gives you a better overall effect size because it's within the same unit and you're not doing any conversions. Because sometimes percentages can be smaller in differences than the actual units. For here, the method, this is how to determine the heterogeneity and how to determine the tau squared, which you'll have to learn about in the book. So Der-Simeonian layered method is the newest method for creating this tau squared, and it is cited as the most robust way to identify heterogeneity. Um, this is the Hertong-Knapp adjustment. This is something that was suggested by the German group that helped write the book. And what this does, it adjusts the effect size based on the number of observations. And this is a really good way to understand if having more observations within a study can account for a lower standard deviation within that study, or if it doesn't, then it'll give you more information about why that wasn't the case. So this is always something good to include. The prediction interval would be like the 95th percent confidence interval, and this is already being shown on the forest plot, so instead of making vertical lines, I just like to show that individually, and I'll run you through how to do that. Even though we do run the prediction, we don't show it on the plot, so that is being negated here with the false command. Also, we have the title listed here. So in our case, this is actually supposed to be mass, but we'll just change that here. So mass and PGR control groups, it can be whatever you want it to be. And then it'll very simply print you the results of your meta-analysis. So here we have a total of 10 studies. Remember, this is different from observations. This is just the number of papers. The number of observations, here we have a total of 239. Observations within our experiment are 141, and within the controls are 98. Within the random effects model, we see the mean difference in the actual units. So in our case, the actual unit difference would be about 0.32 grams bigger because it's a positive number than the control. And the 95th percent confidence interval runs from the low end of 0.1 grams to the high end of 0.54 grams. We can see here that the p-value is significant at the p equals 0.05 or below level. Below this, it has heterogeneity effects that also include the 95th percent confidence intervals. It has the tau squared. It has the tau, which is simply this number that's not squared. And then it has the I-squared statistic. You'll have to learn more about this, and I won't go into what these are calculating in this presentation. But here we do have an overall test of heterogeneity, and since this value is highly significant, we can say that the heterogeneity between studies was significant, which means that not all studies were pointing in the same direction. So from here, this is how to lay out the forest plot. So the forest plot within the meta package is known as forest. And forest reads in the results of your meta-analysis, which was created up here. So remember, this is named result. So the first thing we do is we feed results into the forest plot. In my case, I made this variable called sort variable, and what this does is it takes data, which is our data frame, and it sorts it by the top mean difference, which means that the highest mean difference will be at the bottom of the plot. The layout, revman5, is from there's a package that well, it's more like a standalone software that's called Review Manager, and it got discontinued and people really liked the way that it looked. So when they made this package, they gave us the layout back from Review Manager, and I really like the way it looks as well. This is just some extra stuff to give myself more space, so I added a line underneath it. This is for centering all of the stuff. Digits here determines how many digits each of your summaries will have, so like the means, how many digits do you want them to display. The labels here are the labels for the plot, so either cytokinin treated or untreated. The label on the left would be that if it's to the left of that, left of mean, it'll favor the untreated controls, and if it's to the right, it'll favor the cytokinin. This is for printing the statistics here, so you put true for printing the statistics. And then all of these here are the colors of the overall effect size, diamonds and squares, and these can be whatever you want. Down here is just some stuff for layout and naming some of the things, and then going here as well, it's just the different font sizes. I made this add line down here that formats the heterogeneity statistics to be below the plot, and it looks pretty nice, and I add some spacing here to give them more space at the bottom. So I will run this, and this will generate a forest plot, and then we can see how that looks. So as I've noted, the lowest effect size is listed first, and the greatest effect size is listed at the bottom and to the farthest right. So how to show and interpret these plots is just like in the forest example up there in the text version of it. This is just how to see that in a visualization. So the left-hand side columns are the cytokinin treated, the right-hand columns are untreated, and below them are the means and standard deviations of these different studies. So in our example, random study 7 had a mean in the cytokinin treatment of 2.01 grams, and the untreated had a mean of 2.09, while the standard deviations were 0.11 and 0.19. The total column tells you how many observations were found within the study. So in our case, we had 11 cytokinin treatments and 11 untreated controls. What this did was it gave this study an overall weight of 10%. So this is a pretty balanced study. As you can see, all of the different observations and studies within this meta-analysis have all around 10%, which means that each of them are contributing equally to the overall effect size. So MD, mean difference, and the 95th percent confidence interval is representing the first number, the overall effect size, and then to the right of that, the 95th percent confidence interval. This 95th percent confidence interval, the null effect line is noted here as this black bar that runs at 0. And a good way to think about it is if a effect passes the null effect line, meaning that it goes from negative to positive, such as these three, there is no overall effect, and we can see that here based on the 95th percent confidence interval sticking out and crosses the null effect line. So this means that the p-values of these studies would not be significant from 0 or different from the control. While if we go to study 2, right here, that's the first study that has an overall increased effect size, because that confidence interval does not pass 0. It goes from 0.07 to 0.33. Down here, I have some text that tells you about how to interpret these and how to interpret the heterogeneity measures. So when I send this to you, I'll walk through this and you can read some more information. Now, a good way to understand whether or not your publication meta-analysis has bias or some small study effects, as I note here, you can run something called a funnel plot. And the funnel plot is another function within the meta package. So you just write funnel, you type in your result, and then there's two, you can just put your labels here. So here what we're looking to see is where's the funnel overall. So these are the 95th percent confidence intervals coming off on these pyramids. And we can see that the overall effect is about, 0.27 or zero point 0.3 or something like that. And we're looking for the standard errors to not be all over the place. So we're looking for a nice straight line, which is what this represents here. Another one, what you can look at is called a plot. And what this plot shows is what studies influence the heterogeneity. So here we see all of the standard errors, which is the standard deviation divided by the square root of the number of observations. All of these have a pretty similar standard error. And so this isn't a good way to look at what study is influencing what. And in fact, we don't even have labels on these studies. So we have no way to know what study is contributing what on this plot. So that's what the Bujat plot does. And the Bujat plot shows us that, okay, looks like study 8 actually has way more heterogeneity. And let's confirm that. So study 8 looks like it has, over 100% proportion of heterogeneity. Let's go back up to our forest plot. And study 8, as you can tell, uh, is the most different from the other treatments here. So that's why it's contributing the most to heterogeneity. Because we have three papers that do not cross the null effect line, or that do cross the null effect line, and then these that are kind of all within each other, and then this one's way out there. So that's why it's contributing the most. And this could be interesting to talk about in your results section, because why does study 8 have that biggest difference from the other studies? And it's important to note, maybe it's something to do with the size of the plants, maybe it's the application timing, or if you're doing irrigation or something, maybe the plants were more stressed and so they recovered more with the water application. All right, so in summary, um, keep your question to one sentence and keep your scope tight so you can find the right papers for your meta-analysis. Make sure to document your Prisma with the app that I'll include with you so you can document your search. Create your Excel sheet with the mean, standard deviation, and number of observations as your raw data set. And then really all you have to replace within my example is 5 lines to produce the forest plot. And the process is repeatable for any crop and trait combo, just making sure that you assess the heterogeneity between the studies and you understand the effect of each study on the heterogeneity and overall effect size of your plots. So thanks for listening.